DNA, as you know, been discovered by not accidentally, right? First time DNA was discovered in 1860, right, by Frederick Michel, right? And he is a Swiss chemist. He is the one who discovered DNA first, right? And accidentally then discovered what? Structure of DNA. In between, of course, like we have talked about Griffiths, we have talked about Miller's, right? And then all those tells us that okay, that Hershey and Chase experiment, that everything tells us why DNA is important. And then we started looking at the DNA with lots of different respects, right? And that is where people started working on DNA, and eventually in 1953, DNA structure was discovered. But before that, right? Before that, DNA was, people were trying to crystallize DNA, right? Crystallize DNA. So take DNA in pure form, crystallize that, and obtain X-ray diffraction pattern. And the typical X-ray diffraction pattern, right? One name that we should not forget about is Rosalind Franklin, right? British chemist, right? And then she worked on the DNA crystallographer. So she was a crystallographer. She is known from so many different things, right? From the crystal structure of coal and graphite and things like that, right? So that is where she tried to look into it, and she crystallized DNA for the first time. And in fact, she named her, she was like doing the crystallography and then clicking all those pictures of DNA crystals, right? The X-ray diffraction pattern. And one of that, photo 51, which is famous as this name of place itself, photo 51. So if you Google search photo 51, you will see that typical DNA diffraction pattern. And that, looking at that, right, Watson and Crick could gain an understanding that DNA is nothing but a double helix. It's a ladder and twisted, right? So twisted ladder would look like a double helix, and that is where the DNA double helix started. Like, and then, of course, like that's an iconic and well-sung discovery of DNA double helix, right? And so we will look into how DNA basically is. So DNA, as the name suggests, it's a deoxyribose nucleic acid. Deoxyribose is what? It's a sugar, right? It's a filtered sugar, right? It looks like this. Of course, there is on top of there is an oxygen. Right? This is a pentoid sugar and carbons. So this is carbon 1, 2, 3, 4, and there is a fifth carbon there. Right? So this is how a deoxyribose sugar is. This way you have oxygen in case of RNA, right? And which is not there in DNA. So ribose sugar and deoxyribose sugar. What's the difference? The only difference is oxygen, right, at the second position. So if you remove that oxygen, it would become deoxyribose, and this is the sugar into it. Besides sugar, what is there in DNA? R phosphate group, right? So it's a phosphate that binds to the fifth carbon here, right? And then it connects to the third carbon of the upper sugar molecule. Here, third carbon, right? So that is how it goes. So that's why DNA has the orientation. So if you look into DNA, so if you drop all these kind of details, this is the third carbon, this is the fifth carbon. So that is where phosphate group is attached, right? It is attached to the third carbon here, right? And again another phosphate group, right here, and then it goes like that, right? So it would go upward. Here also, on this third carbon, another phosphate group, right, which will attach here, right, at this sugar, right, and the phosphate group here. So it goes in like that, right, and that is which form the sugar phosphate backbone, right, of the phosphate. Right, so what it formed? The sugar and phosphate backbone of the DNA. So DNA has important parts: sugar, like a phosphate, and to which it attaches the nitrogen bases, like nitrogen base. So that is where it will form so shoot, and then again nitrogen base here, right? This is how one strand of a DNA would look like. Now, as I told you, this is a 5' end. This is 5' end, and this is 3' end. Why? Because it's a 3' third carbon and a 5th carbon. That is where the phosphate group is attached. Now, this nitrogen base is attached to another nitrogen base. In case if we could like just look into this nitrogen base. Nitrogen bases are which? A, T, G and C. Right? And this, they are attached to another nitrogen. C will pair with G. It's always like that, right? G will pair with C. C will pair with A. And A will pair with T. Right? Why there is double bond here and triple bond there? Because they are attached with triple bond and double bond, right, to these nitrogen bases. Now, this nitrogen base is then attached to the sugar here, but now this sugar orientation is reverse. Right, now this sugar orientation is reverse than this. Right, you could see that this oxygen is up, pointing upwards. Here the oxygen is pointing downwards, right? So that is how it always look like. So two strands of DNA are anti-parallel. What do we call them? Anti-parallel. So one goes into this direction, another goes into this direction. So they are anti-parallel. So two strands of DNA, they are attached with the nitrogen bases. Now, very important point here, right? Which is not there in many books. These bonds are what? These are hydrogen bonds. Between two nitrogen bases are attached with hydrogen bonds. And what this attached here are, phosphate group is attached here with, what is the bond between phosphate groups? Any idea? Phosphodiester bond, right? So phosphodiester bond, right? Because this phosphorus is attached here with ester bond and here also with ester bond, so it's a diester bond, right? So two ester bonds that will act. So the backbone is made up of phosphodiester bond, and the nitrogen bases attached with hydrogen bonds, right? Now the hydrogen bonds in the DNA are spontaneously made and break, right? So if you take two DNA strands and then allow them to stand in the tube together, they will find each other's complementary bases. They will bind C to G, G to C, T to A and A to T, and bind together to form the double helix, right? Spontaneously. Don't require any enzyme. It doesn't require any energy for that, right? So spontaneously they will bind together. They will form the double helical DNA. In fact, we can see the same thing happening in RNA too. Right, so RNA is also like, RNA is largely double single stranded, that is what we know about. But then in RNA, right, there are stem and loop structures. So what is this stem and loop? This is where it becomes double stranded, right? Even tRNA, transfer RNA, if you see, you would see these double stranded regions into RNA. Okay, so that is where also you will find the complementary sites and complementary sequences will attach to each other, right? So they will form the complementary bases and attach. If you take DNA for example, right, and just heat it, right, what will happen, right? These strands, the hydrogen bonds in between the nitrogen bases, they will break because of the energy that you give, and the strands will separate. And if you allow it to cool down, right, they would come back again, right? So we call this feature as denaturation of DNA and renaturation. So DNA can denature and renature spontaneously, right? Doesn't require any enzyme, anything like that. However, the phosphodiester bond, if it breaks, right, then you would require an enzyme to come there and put them together. And which enzyme is that? DNA ligase, right? So phospholigase or ligase, right? Why? Because the ligase can alone make this bond, right? Even if you want to break this bond, what would it require? Either you have to take DNA and shake it very heavily, like take DNA and then sonicate it, for example. Then what will happen? The DNA is a small molecule, right? Its diameter is just 2 angstrom, right? So it's like just 2 angstrom diameter. So it's a thin molecule. If you shake it vigorously, what will happen? It will break. It will break in between, right? And then if it breaks, what we call now, there are like lab jargons about it, like lab terminology for that. If I say DNA is broken, DNA is cut, DNA is nicked, right? What does that mean? That means the phosphodiester bond has been broken, right? We say DNA is degraded. Degraded means, or if because you require enzymes to break this phosphodiester bond, we also call it as the DNA is digested. So if you say digested, if you say degraded, right, or if you say broken down, or if you could say that there is a nick in DNA, that means phosphodiester bond has broken down, right? But if I say DNA is melted, DNA is denatured, that means the hydrogen bonds are broken down, right? Understand that? That is a clear distinct how we could distinguish between the making and breaking of these bonds. Remember, right? Phosphodiester bond, you require enzymes to cut, right, or enzymes to join them together. They cannot do spontaneously. If you take two strands of DNA and put them together into a solution and wait for 100 years, they are not going to join together, right? What will happen? You have to use an enzyme to put them together. However, if you denature a DNA, right, and then allow them to renature, cool down. So if you hit it, they would denature. If you cool down, it will renature. They would spontaneously renature. Got a difference, right? So two bonds, hydrogen bonds and phosphodiester bonds. Both have different properties. Both have different designations, and there are different names which we talk about, right? So when we say nick is actually always, right, into, if this is a double-stranded DNA, and if I say there is a nick in it, that means once there is one break in one strand, right? So it's a single-stranded nick, right? If you say if there are then it is a double-stranded nick, right? We have enzymes, right? We call them as restriction enzymes. Right, restriction enzymes, as the name suggests, what? They would restrict the DNA. Restrict मतलब क्या होता है? They would restrict the DNA, mean cut the DNA. Right? So they are like there are enzymes which acts like this. Right? So this is DNA. Right? There are enzymes which act like that. What I have drawn? Caesar. Right? These are molecular Caesars. The enzymes which acts as a molecular Caesar, they are called as restriction enzymes or restriction endonuclease. Right? To be very frank, it's restriction endonuclease. So what restriction endonuclease can do? They can cut the DNA in between. Right? There are other enzymes also. Right? There are enzymes. Like this is a DNA molecule, there are enzymes like here. You know what I am doing? right? Pokemon, you have seen that, right? It chews the DNA from the ends, right? So you have another end, right? There are enzymes which chew DNA from this side also, right? They are exonuclease. What do we call them? Exonuclease. Exo means towards the end, right? Both the ends. So from both the ends, this enzyme exonuclease chew the DNA, right? And then release the nucleotides, right? What do you call this as? Right? Phosphate, sugar, and this unit is called one nucleotide, right? Is one nucleotide. So what exonuclease would do? Will cut and release the nucleotides, right? They would digest DNA completely, right? So nucleotides, of course, DNA is a polymer that you know. It's a polynucleotide. Right? So DNA is called as what? Polynucleotide. A small piece of DNA, what we call it as oligonucleotide, right? So it's a small stretch of DNA, right? So that is where we call it. So nucleotide. So this is nucleotide. What nucleotide would consist of? Sugar, nitrogen base, and phosphate, right? Attached together. So this would be a nucleotide. So the nucleotides make polynucleotide, right? So that is where you must understand that these are the ends, these are the polymers, right? And then those are. Understand this DNA, right? Anti-parallel. Understand that, right? The 5 prime to 3 prime, and this of course 5 prime here for this strand and 3 prime here for this strand, right? So they are anti-parallel, like you are standing on a railway station, right? So what happens? One track is up and one track is down, right? The trains come from this side, the trains come from that side, right? It's like up and down, right? So this is an up strand, this is a down strand. Whatever way you understand or remember that DNA has two strands which are anti-parallel, right? Right. And then two enzymes we talked about are exonuclease and exonuclease and there are endonuclease, right? That is what we talked about. So endonuclease क्या करेगा? Right? It will, but now see the endonuclease are smart enzymes. What is mean by smart enzymes? They can read the DNA actually, right, and cut it, right? Like for example, if you take EcoR1, it will look for the DNA, right, GAATT, CGATT, right, and where it will find GAATT, CGATT, it will go and cut, right? So when it cut, right, it will cut. So REs or restriction endonuclease is a smart enzyme. Why they become smart? I will talk about that down here, right? Right? Whereas exonuclease are what? They chew from the ends, right? Doesn't know any sequence or anything. They just want one end of the DNA. That's it, right? If you get one end, and they are amazingly fast enzymes. Remember, right? We always say that, right? If you have a DNA, if your gel ready to be loaded onto the gel DNA, right? What will happen if the DNA is contaminated with exo or endonucleases? By the time you load that DNA, the DNA is gone, right? Within fractions of seconds they act, right? And they can release the nucleotides of your DNA, right? Now, when you see this DNA, ATGCs and are there. One another thing about that, A will always pair with T, T will always pair with A, G to C and C to G. There is no exception in it. Right? There are few laws in nature, right, that does not have exception. One of that is this, right? That without exception, right, under any circumstances, A will not pair with another A or G or C. Never. It will always pair with T. Right? Right? In no condition, G will pair with C or G will pair with any other than C. They always have that, right? Once we know that, right? Okay. Once this is like look like, right? But then what does that mean? Right? That means DNA everywhere is the same. Right? If you look into a DNA, if I say, okay, this is a gene for insulin and this is gene for hemoglobin, What is the difference between them? Right? These also have the same ATGCs, same phosphodiester group bonds, same hydrogen bonds, right? So chemically it is the same, uniform everywhere, right? In fact, everywhere in the life, right? We talked about origin of life theories. Why we say that life has originated only once? Because you will see that DNA is common in all the life forms. We haven't got any life form which does not have DNA, right? Of course, RNA viruses can be exception, but RNA viruses also has to convert their genome into DNA and then act, right? So that's like HIV is an RNA virus, but then it has to convert itself into DNA before it will can make any effect or make an infection, right? So you have to have that reverse transcriptase enzyme. In fact, in AIDS, one of the mainstay of the treatment is, right, anti-RT, right? That reverse transcriptase. If you have chemical drugs which can inhibit reverse transcriptase, you can stop spread of AIDS virus, right? So that's because, so that means what? Even if it is an RNA virus, right, it will still depend on DNA, right, for any action, right? So in all the life forms, right, that what we know today, right, on this globe, right, everywhere DNA is the genetic material, right? So you can see that, okay, the life forms may be like very different. Like where is an amoeba, an elephant, like maybe a whale and maybe a shark, right, or a bird which flies. There is no commonalities between them, right, as such, apparently, right? But if you look deep down, right, everyone has DNA as their genetic material. So what does that mean? They must have originated from one primordial cell and diversified it down here, right? So DNA proves that theory, that it has. So DNA unifies all the life forms, right, together, right? So it is like, it's like a garland, right? In a garland you have different flowers, right? All size, shape, color, smell different, but one thing common in the garland is that thread, right? That thread runs through all these flowers. DNA is like that, right? So DNA runs as a thread, unifies all these diverse life, right? So that is the most important part of it. Understood? Right? So this is all we must understand about it. Rest of the things, of course, DNA you have been studying since 8th standard, so we didn't want to go into too much of structural details of that. But this is the one thing that you must understand about DNA, right? In order to understand the next part of it. Right? What are the next? Like we have to understand what is a gene, right? Are there genes where and how those are, how they are organized? We'll look into that. But before that, like one important thing that we must talk about DNA is like the DNA makes the chromosomes. Where DNA exists in your cell? In the nucleus, right? DNA is hardly there outside the nucleus, right? There is no DNA outside the nucleus. If you have free DNA in cytoplasm of a cell, there are exonucleases. They would be waiting for it to do their job, right? Eagerly waiting for to do their job, right? They will chew that DNA off within no time, right? So there cannot exist a free DNA in the cytoplasm. So DNA must exist only in the nuclei, right? What happens in bacteria, for example, right? Because that is where we have to speak about, right? Bacteria does not have nuclei, but then their DNA is circular, right? We will speak about prokaryotes and eukaryotes maybe next time when we will meet, because that's very important point, right, into understanding the next thing into your course, right? So that is what we will look into that. But understand that every DNA, every chromosome that you have is one DNA molecule. Every DNA you have is, every chromosome that you have is one complete DNA molecule. So this is a DNA, right? It condenses itself into a chromosome like this, right? A long DNA molecule, right, condenses by help of other proteins. Right? There are proteins like histone proteins that will make it compose into the. So if this DNA replicates, there are two DNA molecules after replication. Now what will happen? They will have a chromosome. They will make a chromosome like this. Right. So every chromosome is what? If you look into, there is one DNA going there. There is one DNA that is going there. So every chromosome is one DNA molecule. So how many such DNAs molecules are there in your genome? Right. So that means where every chromosome that you have is actually one single DNA molecule condensed itself, right, or into a form of chromosome. So chromosome is a structure, right? And the constituent of the chromosome is what? DNA and protein, right? Nothing else, right? There are DNA and proteins that would make a chromosome, right? Understood? So that is how DNA makes your genetic makeup or your genotype. Right? We will talk about those in details down the line.